Welcome back to the deep dive. Today, we are taking a trip into the microscopic world, but not just any part of it.
No, definitely not the nice parts.
Right. We are venturing into the dark, anaerobic tunnels of the human gut and the delicate environment of the urogenital tract. We are looking at the luminal protozoa.
Mm-hmm.
And I know that sounds a bit dense, maybe a little like a dry lab report, but trust me, this is where the biological drama really happens. We're talking about single-celled eukaryotic parasites that have evolved some truly specific and frankly kind of terrifying ways to set up camp inside the human body.
It really is a fascinating area of microbiology. When we talk about luminal protozoa, we're really discussing organisms that have adapted to live in the hollow spaces, the lumens of our intestines and urogenital tracts.
Right.
And, they aren't bacteria. These are eukaryotes. They have nuclei, they have complex organelles, and they have distinct personalities. They're very strategic players in the landscape of infectious disease.
Exactly. And our mission today, for you listening, is to decode Murray's Medical Microbiology, Chapter 72. We're also going to sprinkle in some high-yield insights from Jewett's Chapter 46, specifically focusing on the protozoa. But primarily, we're looking at this through the lens of a medical student preparing for those single best answer questions, or the MCQs. We want to find the patterns, the aha moments.
Because patterns are everything in parasitology. If you try to memorize every single fact in isolation, sizes, shapes, drugs, you will absolutely drown.
It's too much.
It is. But if you understand the biological logic, the strategy of the organism, the answers start to reveal themselves. You stop memorizing and you start predicting.
I love that. Predict, don't just memorize. So to help us organize this, I want to propose a mental model for today's deep dive. I want us to think of this as a chess match.
Okay, a chess match.
Yeah, these bugs aren't just random. They're distinct players with specific moves on the board. We have the invader, the blocker, the opportunist, and the vulnerable traveler.
I like that framework. It categorizes them by their pathogenic mechanism, which is usually how they present clinically to you in a vignette. So the invader would be Entamoeba breaking through tissue. The blocker is Giardia covering the surface. The opportunists are the sporozoa like Cryptosporidium, just waiting for the immune system to drop its guard. And the vulnerable traveler...
That's Trichomonas, the one that can't survive out in the cold, harsh world and needs direct contact.
Right.
But before we meet the individual players, we have to understand the board.
Right.
Murray organizes these guys by how they move. We have 4 groups. And this isn't just trivia, right? The way they move tells us something about their biology.
Absolutely it does. The classification is based on locomotion, and that locomotion is deeply tied to their cellular machinery. First we have the Entamoeba, the amoebae. These move by pseudopods.
Which literally means false feet.
Right.
Which is such a cool biological concept. They don't have legs, they just project their own body forward.
It's cytoplasmic streaming. They extend the actin filaments in their cytoskeleton to push the cell membrane out, and then the rest of the cell just sort of oozes after it.
Like a blob.
Exactly. It's a slow, deliberate, creeping movement. It allows them to navigate through the thick mucus and debris of the gut.
Okay, then we have the flagellates.
Also known as the metamonada in the newer classifications, these move using flagella, which are whip-like tails. Unlike the amoebae, these are active swimmers. They can propel themselves against the flow of intestinal contents. This includes Giardia and Trichomonas.
Yes. Then the ciliates.
The ciliophora, these are covered in tiny hair-like projections called cilia, while a flagellum is like a long whip. Cilia are like thousands of tiny oars.
Beating together.
Beating in a coordinated rhythm, yes. It's incredibly complex machinery for a single cell. The only major human pathogen here is Neoballantidium coli.
And finally, the sporozoa.
Or the epicomplexa. These are the tricky ones. In their mature forms, they don't have obvious locomotion organelles like legs or tails. They are obligate intracellular parasites for at least part of their life cycle.
So they don't swim around?
No, they move by a process called gliding motility, which uses a complex internal motor, but you won't see them swimming across a slide like you would Trichomonas. This includes Cryptosporidium, Cyclospora, and Cystoisospora.
So we have the movement types, but let's dig a bit deeper into the why. Why does a doctor need to know if it's an amoeba or a flagellate? Does it actually change the prescription pad?
It absolutely does. The classification dictates the biology, and the biology dictates the drug targets.
Give me an example.
Well, for example, the ribosomal structure of a ciliate like Ballantidium is different enough that we treat it with tetracycline, whereas the anaerobic metabolism of the amoebae and flagellates makes them prime targets for metronidazole.
Oh, okay.
But even more importantly, this structure helps us understand the cyst versus trophozoite paradigm. And I cannot stress this enough. This is probably the single most important concept for understanding transmission.
Let's unpack that. The cyst versus the trophozoite. I like to think of the cyst as a spacesuit or maybe an armored tank.
That is a very accurate analogy. The cyst is the infectious stage. It is non-motile, but it is incredibly tough. Evolution has designed this form for one specific purpose, and that is survival in a hostile environment.
Outside the body.
Outside the body, yes. It has a thick wall made of chitin or similar polymers that allows it to survive in cold water, in drying soil, and crucially, it allows it to survive the acid bath of the human stomach.
That's the key, right? The stomach is usually our firewall. A pH of 2.0 kills almost everything.
Exactly. The stomach is essentially a sterilization chamber. Most bacteria and delicate cells die instantly. But the cyst just laughs at stomach acid. It passes right through unharmed.
So when you drink contaminated water, you are drinking the spacesuits. You're drinking the cysts.
Precisely. And once that cyst gets past the stomach and enters the alkaline environment of the small intestine, it receives a chemical signal.
What kind of signal?
Usually a combination of the pH change and the presence of bile salts. This triggers excitation, the armor opens up, and the organism emerges.
And what emerges is the trophozoite, the feeding beast.
Yes. The trophozoite is the vegetative motile form. It feeds, it multiplies, and in the case of pathogens, it causes the damage. But here is the major trade-off. It is incredibly fragile.
Because it took off the spacesuit.
Right. It has shed its armor to grow and divide. If a trophozoite is passed in liquid stool and hits the air or dries out or encounters fresh water, it just bursts. It dies very quickly.
This leads to a really interesting diagnostic clue that Murray highlights. If a patient has formed stool, you are likely to see cysts. If the patient has raging diarrhea liquid stool, you see trophozoites. Why is that?
It is entirely about transit time. In a normal bowel movement, the stool dehydrates slowly as it moves through the colon. This dehydration is the environmental signal for the trophozoite to pack up and build a cyst wall.
A process called encystation.
Correct. But if you have diarrhea, the transit is so fast, it's essentially a flush, that the trophozoites are ejected before they have time to put their spacesuits back on.
So liquid stool equals trophozoites, and they die in the environment. Formed stool equals cysts, and they are highly infectious.
Exactly. Which explains why asymptomatic carriers, people with formed stool-passing cysts, are actually much more dangerous for public health transmission than people with active dysentery.
That is a massive public health point. Now, there is one major exception to this rule. The nudist of the group.
Trichomonas vaginalis.
Right. Trichomonas has no cyst form at all. It is always a trophozoite. So knowing what we just said about how fragile trophozoites are, how does Trichomonas survive?
It effectively can't survive in the environment. It can't hang out on a toilet seat for long, and it certainly can't survive in a stream or on food. This biological limitation strictly dictates its epidemiology. It requires direct, intimate mucous membrane to mucous membrane contact to move from one host to another.
That's why Trichomonas is a sexually transmitted infection.
Wow.
Whereas the others are primarily fecal-oral. The biology literally writes the rules of the transmission.
Exactly. It evolved to skip the external environment entirely.
Okay, let's start our chess match. Player 1, the invader.
Right.
We were talking about the amoebae, specifically Entamoeba histolytica.
Entamoeba histolytica is the heavy hitter here. Murray lists several amoeba in the text. Entamoeba coli, Entamoeba hartmanni, and Entamoeba pilecki. But histolytica is the only major pathogen. The others are largely commensals, meaning they just eat bacteria and debris, but they don't attack the host.
This is a huge point for your exams and for real life on the wards. You get a lab report back, right, and it says Entamoeba species seen. You have to know if it's the bad guy or just a bystander. But here's the kicker. Murray says they look identical.
It is a massive diagnostic headache. Entamoeba histolytica looks morphologically identical to Entamoeba dispar and Entamoeba moshkovsky, which are non-pathogenic.
Identical.
Yes, they're the same size, same shape, same nucleus. You absolutely cannot tell them apart by just looking at them on a standard wet mount.
So if they look the same, how do you know you have the killer? There is a smoking gun feature, isn't there? Something histolytica does that the others don't.
There is, and it is a classic high-yield question. If you look at the trophozoa of Entamoeba histolytica under a microscope, you might see red blood cells inside its cytoplasm.
It eats blood.
It eats blood. The name histolytica literally means tissue dissolver. E. dispar and the others do not invade tissue, so they simply don't have access to red blood cells. They just graze on bacteria in the lumen. But histolytica tears into the capillary bed and consumes the RBCs.
Wow.
If you see an amoeba with an RBC inside it, erythrophagocytosis, it is histolytica, and it is deeply invasive.
That is the smoking gun. Ingested RBCs equals histolytica.
Yeah.
Now let's talk about that invasion. You call it tissue dissolving. I really want to understand the demolition equipment here. How does a microscopic blob dissolve human tissue?
It's a highly sophisticated chemical warfare suite. It starts with attachment. The trophozoite has a specific surface protein, a galactose N-acetylgalactosamine lectin. We just call it the galgalinac lectin.
That is quite a mouthful.
It is, but it's critical. This lectin allows the amoeba to lock onto galactose residues on the colonic mucin and the epithelial cells. This is the moment the war is won or lost. If it can't attach, it just gets flushed out.
So attachment is step one. What happens next?
Once it anchors itself, it releases cysteine proteinases. These are enzymes that act like molecular scissors. They specifically target the extracellular matrix, the glue holding our cells together. They dissolve laminin and fibronectin.
So it's literally melting the mortar between the bricks of our gut wall.
Exactly. But it actually gets worse. The host immune system rushes in right neutrophils arrive to fight the infection. You'd think that helps.
Usually, yes. Neutrophils are the cavalry.
In this specific case, they are fuel for the fire. Entamoeba has a mechanism to kill the neutrophil upon contact. And when that neutrophil dies, it explodes, releasing its own toxic enzymes, elastase collagenase, into the surrounding tissue.
So the amoeba tricks our immune system into doing the structural damage for it.
It uses our own soldiers to blow up the bridge. That massive localized inflammation contributes to the formation of the classic flask-shaped ulcer.
Describe that shape. Why is it flask-shaped?
Imagine an Erlenmeyer chemistry flask narrow at the top, wide at the bottom. The amoeba eats through the mucosa, which is relatively tough, creating a small, narrow opening. But once it hits the submucosa, which is much softer and looser tissue, it spreads out laterally.
It follows the path of least resistance.
You're right. So you get this deep ulcer with a narrow neck at the surface and a wide necrotic base underneath.
That's a very vivid image. So clinically, this manifests as intestinal amoebiasis. We are talking about a little watery diarrhea here. We are talking about true dysentery.
Yes. Bloody mucousy diarrhea. Severe abdominal pain cramping, and tenesmus.
Tenesmus is that horrible feeling of needing to pass stool even when the bowel is empty, right?
Yes, caused by the severe inflammation of the rectal nerves. It's incredibly painful.
But the invader doesn't always stop at the gut. Sometimes it breaks all the way into the bloodstream. And where does the blood from the gut go first?
It goes up the portal vein directly to the liver.
And that is where we get the most common extraintestinal manifestation, the amoebic liver abscess.
This is a critical diagnosis not to miss on the wards. It usually presents with fever and right upper quadrant pain. The abscess is most commonly in the right lobe of the liver.
Why the right lobe?
Because the blood flow from the superior mesenteric vein, which drains the right side of the colon, where the amoebas really like to live streams preferentially to the right lobe of the liver.
That's a fantastic anatomical hook to remember. Now we have another food metaphor here. If you stick a needle in that abscess, which, by the way, you usually shouldn't do unless absolutely necessary for impending rupture, what comes out?
Anchovy paste.
Anchovy paste. Why does it look like that?
It's a mix of necrotic hepatocytes, liquefied tissue, and old blood. It has a thick, brown, pasty appearance. Interestingly, and this is a key point for exams, this material is usually bacteriologically sterile.
Sterile?
Yes, it's not pus in the traditional bacterial sense, which is full of living neutrophils and bacteria.
So wait, where are the amoebas? Are they swimming around in the paste?
No. The center of the abscess is dead. The food is gone. The trophozoites are found in the advancing walls of the abscess, eating their way outward into the fresh, healthy liver tissue.
That is such a key detail. The center is dead, the action is at the perimeter. So we have a patient with bloody diarrhea or maybe right upper quadrant pain. How do we definitively diagnose this? We mentioned microscopy is tough because of E-dispar.
Microscopy is the traditional way, but it is insensitive and totally nonspecific. You might miss the trophozoites or confuse histolytica with dispar, so fecal antigen tests that are specific for histolytica are much better now. They detect proteins unique to the actual pathogen.
What about for the liver abscess? If the bug is deep in the liver, will the antigen be in the stool?
Often no. By the time the liver abscess forms, which can be weeks or even months later, the intestinal infection might have completely cleared, so stool microscopy or antigen is often negative in liver abscess cases. This is where serology becomes crucial. Detecting antibodies against Entamoeba in the blood is very sensitive for extraintestinal disease.
Okay, let's talk treatment. This is a vital two-step punch, right? You can't just use one drug.
That is a very common and dangerous pitfall. The primary treatment for invasive disease, the dysentery or the liver abscess, is a tissue agent, usually metronidazole.
Metronidazole, the absolute workhorse of anaerobes. How does it work on a cellular level?
It's a prodrug. The nitro group on the molecule gets reduced by the unique redox machinery of anaerobic organisms, specifically systems like pyruvate ferredoxin oxidor reductase.
PFOR.
Right. This reduction creates toxic free radicals that severely damage the DNA of the parasite, causing it to fragment and die.
But metronidazole has a massive blind spot in this specific context.
It does. It is absorbed so incredibly well by the upper gut that very little of it stays in the actual lumen of the large intestine. It shoots into the blood, goes to the liver, goes to the tissue, so it brilliantly kills the amoebae in the liver and the bowel wall, but it leaves the cysts sitting in the gut lumen completely alive.
So you cure the patient's acute abscess, but they are still carrying the parasite and shedding cysts into the environment.
Exactly. They become an asymptomatic carrier. So you must follow the metronidazole with a luminal agent, something that stays in the gut tube and kills the cysts. The classic choices are paromomycin or iodoquinol.
Tell me about paromomycin.
Paromomycin is an aminoglycoside, but unlike gentamicin or topramycin, it is not absorbed orally at all. It just sweeps through the gut, stays in the poop, and kills the luminal bugs.
So the golden rule is metronidazole to kill the invader, paromomycin to kill the carrier state. Don't skip step 2.
Precisely. If you skip step 2, the patient will either relapse later or silently infect their family.
All right, let's move to player 2 in our chess match, the blocker. We are leaving the world of oozing pseudopods and entering the world of the flagellates. Meet Giardia duodenalis, also known as Giardia lamblia or Giardia intestinalis.
Giardia is a fascinating organism with a very unique aesthetic. Morphologically, the trophozoite is unmistakable under the microscope. It is pear-shaped. It has 2 nuclei that look exactly like large eyes and these parabasal bodies that look like a mustache. It has flagella coming off it like flowing hair.
It looks like a little face looking back at you, or a tennis racket with a face.
It has been described as a sad clown face, which is perhaps appropriate given the utter misery it causes patients.
Now, I called it the blocker because of its mechanism. It doesn't invade tissue like Entamoeba does it. It doesn't eat red blood cells.
Well, Giardia is strictly non-invasive. It stays entirely in the lumen of the small intestine, specifically the duodenum and the jejunum. It has a specialized structure called a ventral sucking disc. Think of it like a powerful muscular suction cup on its belly. It uses this to latch firmly onto the microvilli of the intestinal wall.
It's literally like it's laying down a thick carpet over the gut lining.
That is the perfect analogy. But it's not just passively sitting there taking up space. The physical attachment itself causes microscopic damage. If you have a massive infection, you have millions of these trophozoites suction cupped to the fragile epithelium. This physically causes villus atrophy.
Explain that for us. What exactly is villus atrophy?
Normally, your small intestine is lined with diarrhea, yes. The stool actually floats in the toilet bowl because of the high unabsorbed fat content. And because all those unabsorbed sugars are now moving into the colon, where they are fermented by your normal bacteria, there is a massive amount of gas bloating and flatulence.
The classic purple burps or sulfurous gas patients complain about.
It is remarkably unpleasant for the patient.
Now, who gets Giardia? What's the classic exam profile?
The classic profile is the hiker or camper. Giardia is readily found in clear, cold mountain streams. It has a sylvatic reservoir, meaning it naturally lives in wild animals, particularly beavers and muskrats, hence the famous nickname beaver fever.
So you're out hiking, you see a beautiful, crystal clear stream, you take a drink, and 2 weeks later you have greasy floating stool.
Yes, but it's really not just campers. It is also extremely common in daycare centers because of the highly efficient fecal-oral transmission among diapered children. And there is an important immunological association that shows up on exams all the time, IgA deficiency.
Explain that connection. Why IgA specifically?
Secretory IgA is the main antibody that protects your mucosal surfaces. Its primary job is to bind to incoming pathogens and physically prevent them from attaching to the epithelium. Since Giardia relies entirely on that ventral sucking disc to attach and survive, patients who lack IgA are virtually defenseless. They can't stop the initial attachment.
So patients with selective IgA deficiency are particularly prone to chronic relapsing Giardia infections.
That is a very high-yield clinical connection. IgA deficiency equals chronic Giardia.
How do we diagnose the blocker?
Stool antigen detection is really the best method now. It's highly sensitive and specific. You can do traditional microscopy looking for the sad clown-faced trophozoates or the oval cysts, but Giardia has a frustrating habit of shedding in showers.
Shedding in showers. That sounds hygienic, but I assume it's definitely not.
Not at all. It means the excretion of cysts into the stool is highly irregular. You might have millions of cysts one day and absolutely none the next. So a single negative stool sample does not rule out Giardia. You usually need at least 3 samples collected over different days to be clinically sure.
Got it. And treatment for Giardia.
Metronidazole is the classic choice here as well. Metazoxanide is another very good option, especially for kids. Unlike Entamoeba, you don't typically need a separate luminal agent because Giardia doesn't hide deep in the liver tissue, so the drug levels present in the gut lumen are usually sufficient to clear it completely.
Before we leave the flagellates, let's briefly mention the oddball cousin, Entamoeba fragilis.
Entamoeba. It is a bit of a taxonomical mess, frankly. It is classified as a flagellate based on modern genetics, but under the microscope, it looks exactly like an amoeba. It doesn't have any visible flagella.
Hence the confusing name, Entamoeba.
Exactly. And the fragilis part of the name is because the trophozoites are extremely fragile outside the human body. Interestingly, we have never definitively found a cyst form for Entamoeba.
So how does it get around if it's so fragile and has no cyst? We said Trichomonas solves this by being an STI, but Entamoeba is an intestinal bug.
That is the great mystery. The leading scientific theory is the Trojan horse hypothesis. It is thought to travel safely hidden inside the eggs of Enterobius vermicularis, the common pinworm.
That is wild. It hitchhikes inside a worm egg to get safely through the stomach acid.
That's the working theory. The tough pinworm eggshell acts as the spacesuit for the fragile Entamoeba. It is a very strong clinical association. If you find Entamoeba in a patient, you should immediately look for pinworms and vice versa.
Okay, moving on to player 3 on the board. We are still in the flagellate family, but we are leaving the GI tract and heading south to the urogenital tract. This is the vulnerable traveler Trichomonas vaginalis.
Trichomonas is the most common non-viral sexually transmitted infection worldwide. As we mentioned earlier, it has no cyst form. It is a trophozoite, full stop.
And what does it look like?
It is pear-shaped. It has 4 anterior flagella and a very characteristic undulating membrane, like a fin that ripples down its side. It is highly motile. On a fresh wet mount, you see it rapidly twitching and jerking around the slide.
The twitching motility is the absolute key buzzword for the boards. Now, clinically, this presents very differently in men and women.
Yes. In men, it is usually completely asymptomatic. They are silent carriers. The parasite lives in the urethra or the prostate, usually causing no symptoms, though it can occasionally cause a mild non-gonococcal urethritis. In women, however, it causes a very symptomatic vaginitis.
And it has a distinct presentation. We talked about anchovy paste for Entamoeba. What is the food metaphor for the cervix in Trichomonas?
The strawberry cervix.
The strawberry cervix.
It refers to punctate hemorrhagic spots on the cervix, medically known as colitis macularis. It looks literally like the surface of a strawberry. It's a classic clinical sign, though realistically you only see it in about maybe 2 to 5% of patients with the naked eye. It's much more visible if you're using colposcopy.
And what about the vaginal discharge?
The discharge is highly characteristic. It is frothy, yellow-green, and very foul-smelling.
Frothy is the key word there. Why is it frothy?
It's due to gas production by the organism's metabolism. If you're comparing vaginitis causes, Candida is thick cottage cheese. Bacterial vaginosis is thin and gray. Trichomonas is frothy and yellow-green. And there is a critical pH clue here, too.
Let's talk about the pH.
Yes. The normal healthy vaginal pH is acidic, usually less than 4.5. This protective acidity is maintained by normal lactobacilli. In Trichomonas and also in bacterial vaginosis, the pH goes up. It is typically greater than 4.5. This reliably differentiates it from a yeast infection where the pH remains normal and acidic.
So pH greater than 4.5 plus frothy discharge equals Trichomonas. How do we confirm the diagnosis?
You can do a wet mount. You take a swab, put it in saline, look for the twitching bugs under the microscope. It's cheap and it's fast, but the sensitivity is quite low, only about 60%. You miss a lot of active cases.
So if the wet mount is negative, but clinically you still really suspect it?
NITIS nucleic acid amplification tests are the absolute gold standard now. They are highly sensitive and specific.
Treatment. This is crucial because of the epidemiology we discussed.
Metronidazole again, or tinidazole. But the absolute most most important part of treatment is the ping pong rule.
Explain the ping pong rule for us.
Because men are usually asymptomatic carriers, if a woman is treated and cured but her male partner is not, he will simply reinfect her the very next time they have sex. The infection bounces back and forth like a ping pong ball. You must treat the sexual partner simultaneously, regardless of whether they have symptoms. Expedited partner therapy is key here.
Excellent.
Ooh.
Let's move to player 4. The giant. The ciliate. Neoballantidium coli.
Formerly known as Ballantidium coli, this is the largest protozoan parasite of humans by a wide margin. It is absolutely huge compared to the others.
Because how huge are we talking?
The trophozoates can be up to 200 micrometers long. To put that in perspective, a normal red blood cell is about 7 micrometers. So this is a monster. You can almost see it swimming with a low-power magnifying lens.
It's the Godzilla of protozoa, and it has a very specific animal reservoir.
Pigs. This is primarily a disease of swine. Humans are just accidental hosts. So the classic exam case history is a pig farmer or someone handling raw pork products who develops acute diarrhea.
And clinically, it acts a lot like Entamoeba.
Yes, it is deeply invasive. It has a cytostome, a literal cell mouth, and it releases hyaluronidase enzymes. It burrows directly into the gut wall, causing deep ulcers and bloody dysentery. But unlike Entamoeba, it rarely goes to the liver. It tends to stay locally in the gut mucosa.
And under the microscope, besides just being huge, it has a very distinguishing internal feature.
A large kidney bean-shaped macronucleus. Ciliates are biologically unique because they have 2 distinct nuclei, a micronucleus used for sexual reproduction and a massive macronucleus for daily cellular operations. That macronucleus is massive and incredibly easy to spot.
Diagnosis is generally easy just because of the sheer size. But treatment gives us a slight curveball. We've been saying metronidazole for everyone else.
This is where Neoballantidium significantly differs. The drug of choice is actually tetracycline.
Tetracycline. But that's usually an antibacterial for things like chlamydia or mycelia. Why use it for a giant protozoan?
It comes down to the structure of the ribosome. Tetracyclines work by inhibiting the 30S ribosomal subunit. It turns out that the mitochondrial ribosomes of Ballantidium are uniquely sensitive to this specific blockade in a way that the amoeba simply aren't. It's a great clinical reminder that even though we loosely group these things as gut bugs, their internal molecular machinery can be vastly different.
The formula is pig farmer plus dysentery plus kidney bean nucleus equals Ballantidium, which means you prescribe tetracycline.
That is the perfect summary.
Okay, we are entering the final group. Player 5, the opportunists. The sporozoa, specifically the coccidia. I call them the acid-fast club.
This is a very important clinical distinction. These organisms, Cryptosporidium cyclospora and Cystoisospora, share a unique staining characteristic with tuberculosis. Their oocysts have a thick, waxy wall that stubbornly retains the red carbylfutzin dye in a modified acid-fast stain. They turn bright pink or red against a blue background. This is the key way labs find them in stool, especially when you are working up a patient with chronic, unexplained diarrhea.
Let's start with the most famous one of the bunch, Cryptosporidium.
Cryptosporidium is an absolute master of environmental survival. Its oocysts are incredibly tough. They are highly resistant to standard chlorine levels.
This is the famous Milwaukee story, right? This is the one waterborne outbreak every medical student learns about.
Exactly right. In 1993, Milwaukee had the largest documented waterborne outbreak in U.S. history. Over 400,000 people got sick. The city water supply was properly chlorinated, but Crypto didn't care at all. The oocysts passed right through the chemical treatment plant. It actively requires fine physical filtration to remove it from water sources because the cysts are so small and chlorine resistant.
So if you see a board question about a swimming pool outbreak, because remember, chlorine doesn't kill it, or a municipal water supply failure, you immediately think crypto. Now, the clinical picture here depends entirely on the host's immune status. It's really a tale of 2 diseases.
Right. In an immunocompetent person, you or me, it causes a profuse, watery diarrhea that lasts about 10 to 14 days. It's intensely unpleasant, but it is self-limiting. Your immune system clears it, and you get better on your own.
But in an immunocompromised patient, specifically someone with advanced AIDS,
it is devastating. It causes a severe cholera-like diarrhea. Patients can lose liters of fluid a day, up to 10 or 20 liters in severe cases. It is chronic, deeply wasting, and can easily be fatal due to profound dehydration and electrolyte imbalance.
Murray mentions a weird biological quirk about exactly where crypto sits in the gut lining. We talked about Giardia being on top and Entamoeba being deep inside. Where is crypto?
It is intracellular, but extracytoplasmic.
Which honestly sounds like a complete contradiction. How can you be inside but outside?
Think of it like someone getting into bed and pulling the sheet tightly over their head but staying entirely on top of the mattress. The parasite attaches to the brush border, and the host epithelial cell membrane actually grows up and completely over it, enveloping it. So it is hidden from the gut lumen under the sheet, but it never actually enters the deep cytoplasm of the mattress. It sits in this unique little parasitophorous vacuole right at the surface.
That is a brilliant mental image. Little hat sitting on the villi. How about diagnosis?
Modified acid-fast stain. You see very small red dots, about 5 to 7 micrometers in diameter. Or more commonly now, you use antigen detection via EIA or immunochromatography, which is much more sensitive.
And treatment, because it's notoriously tricky for the AIDS patients.
For immunocompetent people, nitazoxanide is the drug of choice. It interferes with the electron transfer reaction, essential for anaerobic metabolism. But for AIDS patients, the antiparasitic drugs really don't work very well. You can give nitazoxanide, but it rarely clears the infection completely.
So what do you do for them?
The only real definitive treatment is to fix the underlying immune system. You must initiate antiretroviral therapy, or RT. As the patient's CD4 count goes up and the immune system finally comes back online, the body can finally clear the parasite itself.
Next, in the acid-fast club, we have Cyclospora cayetanensis.
Cyclospora is clinically very similar to Crypto, but there are 4 key differences you need to memorize for the exam. First, it is physically larger. The oocysts are 8 to 10 micrometers, which is about twice the size of Crypto.
So big red dots versus little red dots.
Correct. Second, the acid-fast staining is highly variable. Some look dark pink, some light red, some are totally clear. It has a mottled appearance on the slide. Third, and this is a great trivia point, it has a cool trick called autofluorescence. If you put the slide under UV light, the cysts naturally glow blue-green. Crypto absolutely doesn't do that.
That is a great exam differentiator. Autofluorescent oocysts equals Cyclospora. And the epidemiology is quite different, too. Crypto is often water or pools. Cyclospora is
raspberries, or basil, or mesclun lettuce. It is famously associated with imported fresh produce. Large foodborne outbreaks are very common in the summer.
And the treatment is different, too, right? You don't use nitazoxanide.
No, you don't. For Cyclospora, the definitive treatment is trimethoprim sulfamethoxazole, also known as Bactrim. It works very well by inhibiting the parasite's folate synthesis pathway.
Finally, rounding out the group, Cystoisospora belli, formerly known as Isospora.
This one is the football. The oocysts are huge, oval, and distinctly football-shaped. Much bigger than the others, usually 20 to 30 micrometers. It causes severe malabsorption, similar to Giardia, but typically presents in AIDS patients.
And treatment for the football.
Same as Cyclospora, you use TMP, S, and Z.
Okay, we have covered the entire board. Let's do a quick rapid-fire synthesis before we hit the quiz.
Sounds good.
If my patient has bloody diarrhea, who are my main protozoal suspects?
Entamoeba histolytica, look for RBC ingestion, or Neoballantidium coli, look for pig exposure and giant size.
If I have a patient with watery, greasy, floating, foul-smelling diarrhea...
Giardia, the blocker. Think malabsorption and campers.
If I have severe watery diarrhea in an AIDS patient...
Cryptosporidium, think chlorine-resistant, small acid-fast dots. Or Cystosospora, the large footballs. Or possibly Cyclospora.
If I have a patient with frothy vaginal discharge.
Trichomonas. Look for the strawberry cervix and an elevated pH greater than 4.5.
And a quick treatment cheat sheet. Entamoeba.
Metronidazole plus paramomycin. Never forget the luminal agent.
Giardia and Trichomonas.
Just metronidazole.
Cyclospora and Cystoisospora.
TMP-SMZ.
Cryptosporidium.
Nitazoxanide if they are healthy, or ART if they have AIDS.
Fantastic. Now let's put you, the listener, in the hot seat. It's time for rapid recall. I'm going to give you a short clinical vignette. I'll pause for a second for you to think, and then our expert will give you the answer and the why.
I'm ready.
Question 1. A 30-year-old male returns from a camping trip in the Rockies. He complains of foul-smelling, greasy diarrhea that floats in the toilet bowl. He has lost 5 pounds, but has no fever and no blood in the stool. Stool microscopy shows a pear-shaped organism with 2 prominent nuclei. What is the mechanism of his diarrhea?
The answer is villus atrophy and malabsorption. This is a classic Giardia case. The clues are the camping history, beaver fever, the greasy floating stool indicating statorrhea, and the face-like trophozoite. The mechanism is strictly non-invasive. It just attaches via sucking disc, damages the microvilli, and actively prevents fat absorption.
Question 2. A patient presents with right upper quadrant abdominal pain and a fever. Imaging shows a large, distinct lesion in the right lobe of the liver. An aspirate is performed yielding a brown, thick fluid described as anchovy paste. What characteristic of the stool finding would definitively confirm the causative agent?
The answer is trophozoites containing ingested red blood cells. This is an amoebic liver abscess caused by Entamoeba histolytica. The pathognomonic feature of invasive E. histolytica is the erythrophagocytosis, the ingestion of RBCs. Remember, the anchovy paste abscess fluid itself is often completely sterile of organisms they are hiding in the active walls.
Question 3. An HIV-positive patient with a CD4 count of 50 presents with severe, intractable watery diarrhea. A modified acid-fast stain of the stool reveals spherical red organisms measuring roughly 5 to 7 micrometers. Which specific water treatment method would have best prevented this infection?
The answer is fine filtration. This is Cryptosporidium. The strong clues are the advanced stage status, the severe watery diarrhea, and the very small 5 to 7 micron acid-fast leukocysts. Standard municipal chlorination does not kill Cryptosporidium cysts. They are highly resistant to it. You must physically filter the water.
Question 4. A 24-year-old woman complains of severe itching and a frothy yellowish vaginal discharge. On physical exam, the cervix has visible punctate hemorrhages. The vaginal pH is tested and is 5.5. Which specific form of the parasite is responsible for transmission to her partner?
The answer is the trophozoite. This is Trichomonas vaginalis. The clues are the frothy discharge, the strawberry cervix, and the elevated pH above 4.5. Trichomonas is biologically unique because it has absolutely no cyst form. Transmission is exclusively via the fragile trophozoite during direct intimate sexual contact.
Question 5. Several guests at a summer wedding reception suddenly developed profuse watery diarrhea. The outbreak is epidemiologically traced to a salad containing imported raspberries. Stool testing reveals an organism that is acid-fast but also autofluoresces under UV light. What is the pharmacological treatment of choice?
The answer is trimethoprim sulfamethoxazole, or TMP-SMZ. This is Cyclospora chiatinensis. The clues are the imported raspberries indicating a foodborne source, the acid-fast staining, and crucially the autofluorescence, and the slightly larger size compared to Crypto. The treatment is Bactrim, not metazoxanide.
Question 6. A commercial pig farmer presents to the clinic with abdominal pain and bloody dysentery. Stool microscopy readily reveals a very large motile parasite covered entirely in cilia with a prominent kidney bean-shaped internal nucleus. What is the organism?
The answer is Neobalentidium coli. The definitive clues are the occupational pig exposure, the dysentery meaning tissue invasion, the huge microscopic size, and that classic kidney bean macronucleus.
How did you do? Hopefully those patterns are really starting to stick in your mind.
And if you missed any, don't worry, just go back to the specific section in Murray chapter 72. Look closely at the textbook tables comparing the cyst sizes and the morphology. Those visual anchors in the text are really helpful for exam recall.
As we wrap up today's deep dive, I want to leave you with a provocative thought. We spent a lot of time today firmly distinguishing between pathogen and commensal. We confidently say Entamoeba histolytica is bad and Entamoeba coli is good, or at least neutral.
But the clinical line is getting much blurrier lately. We briefly mentioned blastocystis and Entamoeba. For years, we confidently thought they were just harmless commensals. Now we think they might actively cause chronic disease in some people, but totally spare others.
Why is that? Is it a specific hypervirulent strain of the parasite? Or is it the terrain of the host? Does your specific baseline microbiome composition determine whether blastocystis makes you sick or just hangs out peacefully?
It really raises the fascinating question of whether pathogenicity is a strict intrinsic property of the bug or if it's actually a dynamic relationship between the bug and the host's unique ecosystem. As we learn more about the gut microbiome, our rigid definitions of parasite versus commensal might really have to evolve.
Definitely something to chew on. But in the meantime, please wash your imported raspberries, aggressively filter your mountain water, and if you are going camping, watch out for the beavers.
And if you prescribe metronidazole for amoebiasis, please don't forget the paramomycin.
Amen to that. Thanks for joining us on this deep dive. Keep reading, keep predicting, and we'll see you next time.